think we are live now. I would like to welcome everybody, especially Jennifer, our speaker today. We had some minor technical problems regarding connection with Jenny has an Apple computer, and apparently only Chrome that works with this software that we are using. Anyway, I just first, so what will happen today is that I'm going I'm sharing the presentation, so Jenny will ask me to go forward and backwards, and I think it will work fine. Just before we start, I just need to say a few words to the audience. So please remember that you need to sign a list with your name by the end of the talk. This is important for your certificates, and also the questions. You can write your question in the chat. What else? We do have a WhatsApp group that we also put the link in a moment here. That's important. Everybody is welcome to join us. There we can share a lot of information on proteomics and also about these webinars. So without further ado, I would like to give the time now to Gilberto, because he's going to present Jennifer for us. Thank you.
Okay, thank you, Tiago. Everybody's hearing me?
Yes.
Okay. Yes. Yeah, I need a feedback. Okay, Brazilians, welcome everybody. Students, researchers, seniors, recent doctors, postdocs, masters, PhD students, undergraduates. I know you have a beautiful presentation today. I'll introduce Dr. Jennifer. She has an extensive and I would say outstanding scientific life. As I told you last time, we had Susan Weintraub, we have Dr. Jennifer Van Eyck now, and we have Dr. Solange Serrano. They are outstanding women in science. I hope they be examples of their work to inspire young ladies, Brazilian scientists. I'll highlight quickly some of her achievements. She has a very long CV, but I'll shorten it. I hope Jenny doesn't be upset with that. She has a BSc in biology and chemistry at the University of Waterloo in Canada, then a PhD in biochemistry at the University of Alberta in Canada. Then she has postdoc works in Germany. And finally, she worked also at the University of Illinois in Chicago, USA. Well, now she is a director of the Advanced Clinical Biosystems Research Institute, as well as professor of medicine at Cedars-Sinai Medical Center in California. She has been highly honored also. For instance, she standard in 2020 as one of the top analytical scientists power lists in 2020, I said. She received also in 2020 the National Academy of Sciences Presidential Award, the International Society of Heart Researcher, many others also awards like Donald Hunt. Donald Hunt is one of the pioneers in the use of mass spectrometry for the study of proteins. A distinguished achievement in proteomics award granted by the United States Human Proteome Organization, but also the International Human Proteome Organization granted to Jenny the distinguished achievement in proteomic science, the award. To finish it quickly, I want to say a little bit about her contributions to science. She has 26 patents or patent applications, over 300 articles and reviews, plus 13 editorials, plus 24 chapters, plus 2 books. And highlighting also 2 research fields that Dr. Van Eyck is involved today in proteomics of cardiovascular diseases and COVID-19. In cardiovascular diseases, she is mostly involved in disease-induced modifications of CTNL, which is troponin, and cardiac myofilament proteins, always in diagnostic value. I mean, she's direct also to clinical proteomics. And the COVID papers and data analysis also, methods developed. And she began with colleagues. She's a founding member, I would say, of a group of clinicians and investigators who initiated coronavirus risk associations and longitudinal evaluation, focusing COVID-19 papers and method development for data analysis. Jenny, I think you can take over now, and I hope you enjoy staying with us all this time you spent. Okay, thank you very much for accepting our invitation. We love it.
It's my absolute pleasure to be here today and to talk to everyone. And it's my pleasure to know so many of the Brazilian group that's working in proteomics. And I hope to meet the rest of you soon and one of these days be able to interact more. You'll see I'm a little bit passionate about proteomics and about the work that we do. And actually, we just had this conversation this morning in our lab about how challenging proteomics still remains, even though we've made such headway. And it's only by working together can we actually make it to be what it can be and to really change medicine. So that's what we're going to chat about today, really about the future of proteomics in the context to personalized medicine and what we're thinking about here at Cedars-Sinai. And what we're trying to put into place so that we can really get proteomics discoveries into the clinic, but also to use proteomics in the clinic itself and how we're thinking about it. And what's fun is other people are thinking about it differently, and together maybe we'll get there. So if we can go to the next slide. The next slide is really talking about what are the challenges in precision medicine or personalized medicine or individualized medicine. People call it all different things. And I think what it really is that there's 2 parts to it. One is the diagnostic side, and certainly our lab has done a lot of work to, and still continues to do a lot of work to try to get biomarkers into the clinic. And I'll talk a little bit about that. But there's this other side. You could have the best diagnostic markers in the world, but if we don't have therapies, then we're stuck. So to me, we have to move proteomics not just to be impacting efficiently getting biomarkers to the clinic and diagnostic uses. We need to find a way where proteomics can really change the paradigm of how we think about drug development. And not only that, how we change from developing a drug to actually this concept of individualized therapies. So we're going to talk about that. So I think there's definitely a growing need for new approaches or new concepts in the area of personalized health. I think treating an individual disease based on their proteotype, based on their proteome, based on the proteome and phenotype together is a way forward. And of course, the phenotype includes genetics. That is your background. But we all know the proteome is also changing very quickly and can represent more the diversity of the chemistry and the biology and physiology that's going on in an individual. And then we have to get to this concept from being able to diagnose to being able to treat the right disease with the right pathway. And I learned that lesson several years ago when we were studying some well-known drugs to a kinase. Everyone thought they knew what was going on. And then we realized the clinical trials in patients were failing because they hadn't realized that there was a different isoform of the target drug, of the target to which the drug was made. And it wasn't going to that target. They were targeting the wrong pathway, even though they got the kinase right. How the kinase worked was different. Excuse me. Can we go to the next slide? So we want to talk today about this paradigm shift. And it comes from this concept that, even today we say we have a drug and find me patients. And we want to move that from that centricness to being able to say we have a patient and what would be that right treatment for them. recognizing that the patient isn't just sick because of one thing, but because they have a spectrum. They may be diabetic, they may not be. They may smoke, they may not. They may be obese, they may not. They may have a genetic preservation towards something, or they may not. It is that complexity to which we need to do a treatment. So if we go to the next slide, what it shows you is that what we do currently today is to look for subclassification. We ask for biomarkers that allow us to subclassify an individual so we could use that drug. You see that a lot in cancer. We want to have certain genetic backgrounds in a person before we will treat them for with a certain drug in cancer. Now, what we want to do, that's subclassification, is we want to move the paradigm to saying we want to know you so that we can prescribe the right drug. So if you have breast cancer today, you might have 4 different drugs you could be on, and they just start you with drug 1, then they go to drug 2 if drug 1 doesn't work. Well, we want to be able to do it to know which drug we should do for you first and start with it, as opposed to just doing a regimen that was defined based on subclassification. So it's really a twisting of what we do now with the tools we have now in order to really focus it on the individual and on the patient and recognize their complexity. So if we go to the next slide. So we're moving really to personalized diagnostic and individualized therapies. Those are the 2 terms to keep in mind. So if we go to the next slide, we're going to start first on the biomarker part, and then we're going to move to what we're doing in individualized therapies a little bit later at the back end of the talk. So personalized biomarkers, I can tell you it's challenging. To really go from discovery, I think we're pretty good at doing that now, but to take that all the way through till you have a clinical utility established, clinicians are willing to use it, it becomes defined, and it's actually in a clinical chemistry lab as a lab developed test or through the FDA if you want to sell it. So we really want to be able to have these biomarkers that allow us to understand you, and not just necessarily one disease, but the spectrum of you. So we need to move from this idea of one disease to this holistic idea that you're more than just a disease. I firmly believe that we need to move from a single time point to multiple time points. And then we have this thing called telehealth, where you don't actually have to see a physician, that we can actually understand your biochemistry and your phenotype and how you're responding to therapy. And you don't necessarily have to do a face-to-face. And then we want to have this increasing efficiency to move biomarkers to the clinic. So that's what the challenge we believe we have to deal with. So we go to the next slide. The paradigm shift from one marker to understanding that it's holistic. So if you go to the next slide, I think we have an example of CRP as an example of a single biomarker. Great biomarker for inflammation. We use it clinically, not so much, although it can be ordered, because it really is a generic inflammatory marker. It's an acute phase marker, great marker up in many, many diseases, but it really represents one part. But of course, an individual is many things. So we recognize that disease is not necessarily represented by a single protein, but rather a network of interconnecting proteins. And you'll even see that later when I talk about acute phase proteins. And that during your disease development, those pathways may change. And so if we have assays that are able to monitor these different pathways, maybe we can better define you. But of course, this is dynamic, so you need to have longitudinal measurement, and then we need to be able to understand how to understand how these different pathways work. So if we go to the next slide, we have several assays. One, of course, is a targeted based mechanistic assays. That's what we try to do. We have 2 big ones. One is an acute phase protein assay, and the other one is our health surveillance assay, which represents 60 proteins for pathological signature and is really great at monitoring what an individual's doing. And we can use this on plasma, on remote sampling devices across the board, really as a way of doing thousands and thousands and thousands of samples per day, per year. These are made to be high throughput, super accurate, very, the coefficient of variance is well under 20%, if not well under 5%. These are high-end preclinical assays made to be done in large-scale population studies. So if we go to the next, but of course our idea is to get those into the clinic. So what can you measure on this diversity of the proteome, knowing that you can have the gene products, which then can undergo splice variants, so you can have isoforms, and all the way to having a host of post-translational modifications, many of which we still as proteomics do not routinely measure. So the exponential space, chemical space to which the proteome does is huge. And so one way to be able to do this is, of course, by mass spectrometry. And the way we do it is we digest, for the most part, the proteins into little peptides. Why? Because mass spectrometers are more accurate and precise the smaller the molecule. So peptides, they're more accurate in measuring. And we measure them. And then we use bioinformatics and computational tools to put the jigsaw together. But what we're measuring and quantifying are peptides. And it's those peptides that we assume roll up to represent a protein, or a protein with a post-translational modification. And so the picture of above, I got from Mike McCoss, because I always liked it, your beautiful high resolution, the Mona Lisa. And when we put together all our proteome, we get a kind of a fuzzy picture of Mona Lisa, but we can still tell it's Mona Lisa. And that's what we're trying to do when we do discovery. We try to see the whole thing. When we're doing targeted, we may only choose to look at her eye, her fingers, her part of her hair, or the background, representing different parts of it, each of them representing her face, her hands, or the trees behind her. So again, discovery helps us to see kind of fuzzy, but when we do targeted, it's a very precise but incomplete picture. So you have to be very careful what you do. So we go to the next slide. Along the route to biomarker discovery, we often, of course, do discovery, trying to find those analytes that are changing. And what we've learned over the years, and it's been many years and many developments of pipelines, always trying to get it more robust and more throughput, is the higher the number of patients you have in your cohort for discovery, the more likely you are to be right at the end of this very long journey. And I know I often hear that the precision and accuracy that we demand on our targeted assay, on our discovery assays, is unrealistic, and that we should be okay with kind of fuzzy data on the discovery, because you are going to go through this more targeted, higher throughput journey towards clinical utility and towards clinical assay. I think in theory, that sounds awesome. But the practice of it, to make a high quality targeted assay, to make a targeted assay that can run 100 samples is not the problem. The problem is making a clinical targeted assay that can be run on 10,000 samples, or can be run on 20 samples today, 20 samples next week, 20 samples a year from now, and you have the exact same value. That's what you need to establish clinical utility. That's what you need in order to allow you to get an assay to go to clinic. And so that workload is really so great that it's worth the effort to make your discovery assay bigger and more precise. So we work hard. It's not, obviously the discovery assays are not even close to the robustness of the clinical assays that we do, but we sure try hard to make that to happen. So DIAMS is what we use primarily in our discovery for plasma. And then we're going to compare it to the acute phase assay, which is an immuno-SysCapa assay versus something less like CRP. So if we go to the next one, underneath it, we have developed something called the Precision Biomarker Labs. It is 3 new labs. They were developed just last year. There's a CRO, which does targeted assays and discovery assays for clients. There's a translational lab that's focused on these production of these targeted assays that are really high-level preclinical. And then we have a CAP-CLIA clinical chemistry lab where we can actually release clinical data from our mass spectrometry assay. 3 labs together to try to bring all that expertise to help in this translation. So one of the fundamental things for our lab as we start to scale everything is really in the automation of sample prep. And we started this journey a long time ago. It was at Hopkins, so it's at least 8 years ago, before people were thinking about it in the proteomics. We started with Beckman Coulter and SCIEX on the NPX, and now we're on the i7. That's a picture of the i7. It is a dual arm, large automation system, but it allows us to go everywhere from sample denaturation, reduction and alkylation of your cysteines, to digestion, to online desalting. that whole thing can be done automated on the i7 system. You can now buy the same system. We programmed it. It's available through Beckman Coulter, but it was transformative, not only in that we were able to run 96 well plates, and we're hoping to move to the 384 well plates soon in the next year, but it allows us to really reduce our reproducibility. It allows us to control many aspects. So essentially, we put the sample on to 96 well plate. It goes on the automation system, and then you essentially pick up your plate at the end, and it's ready to go onto the mass spectrometer. And we have several publications on that, but it really was transformative for our lab. As I mentioned, if you go to the next slide, there's a little black box at the bottom. If you actually hit that little black box, you'll be able to actually see the automation system in place. So if you hit that black box, we should be able to see the little video. There's the i7 doing its really cool thing, because you can program it to do all kinds of stuff. And essentially, we just put out into bioarchives, the papers in that clinical chemistry right now, a workflow that can do depleted plasma using camel antibodies, so antibodies made in camel, so they're single chain antibodies plus whole blood remote sampling devices, where we have a common pipeline that can take it all the way through to finally the desalting onto DIA, and then we have QC backup that's all automated on our mass spectrometers. And essentially this has allowed us to be able to really do higher throughput, like large numbers of samples. And so the goal is to be able to do that. So if we go to the next slide, what do we see with that? Well, here what you're seeing is MS intensity, so kind of concentration across a number of proteins. This isn't the proteins we observed. Obviously, they're proteins we observed, but it's not all of them. Those numbers are massive. What these proteins represent are proteins that are seen when we do a 5 by 5 by 5, which is 5 samples done 5 times, prepared per day for 5 days. So this is really looking at robustness of each peptide and each protein over time. And you can see the standard deviation. There are some high abundant proteins that have larger standard deviations, and some low abundant proteins that have almost no deviation. And so we have to figure it out. And what you can see is the 3 different methods. So green is naive plasma. This is on a 21-minute gradient on an EvoSep. So it's a 25-minute turnaround time. It's 60 samples a day. You can see in after depletion in red, you get more proteins. They slightly overlap, obviously, with naive, but together you actually get the biggest proteome coverage. That's in gray. And then you have the beautiful blood proteome, which is in blue. So what you can see is we get coverage. We can see proteins like CRP, TNF alpha. It is dependent, the total number is dependent on the disease. This is healthy individuals. So it's, kind of the smallest proteome that we normally see when compared to other ones. But we can accurately measure about 400 proteins using 2 mass spectrometers on a 21-minute gradient, so 60 samples per day. We're able to accurately measure with coefficients of variance of under 20% around 400 proteins. Now that does change, as I said, with diseases, but you can see here for CRP, the peptides that represent CRP, you roll them together. This is healthy, COVID positive and COVID negative hospitalized individuals. You can see the measurements that we're getting is around 200 individuals in there in each group, and you can see that very well. So you can get tight measurements on it. And there's a picture, of course, of the Mitra devices, which are what we use a lot to get these remote sampling, where you can pinprick your finger and mail it to us. So it's a beautiful proteome to work with. It's our favorite, actually. Plasma and depleted plasma are okay, but we really like blood. It's a great matrix to work with. So if we go to the next slide, this is kind of an outline of the pathways. I think that's, to me, what's more important than the actual protein. And you can see the list of some of the pathways that we can see, the number of proteins we see in each method. So the 21-minute gradient. The middle one is the 21-minute gradient done both with depleted and naive. So it's our broadest coverage. And then the other one, which we often use, is naive for 60 minutes. So if you want to just run one gradient, a 60-minute gradient is really well done. And you can see that for the naive. And so you get more coverage of pathways or more representation per pathway as you go to longer flow rates. But the question always becomes, what do you need to see? People assume we need to see low abundant proteins, but that's not true all the time. It really depends on what you're asking and what your hypothesis is. So if you go to the next slide, please. So we in the Corral in COVID-19, we at Cedars started something called the Corral. It was actually Susan Chen and I and Kimia Sabani, who's a clinical director of clinical chem. We decided to create this really large initial tracking of COVID-19 individuals. And we call it Corral because we started it in March, right after on March 14th, when there was this incredible viral video in Italy where all the people, Italians in Rome, were singing on their balconies in support of their healthcare workers. And it was so touching that we decided to name our really large consortium. It's many, many studies under the Corelli brand name, and we went with this. I'm going to talk a little bit about the Corelli study. And one of our hypotheses, what the severity of the COVID-19 response will be correlated to both the extent of immune modulatory as well as other detrimental systemic responses. So it's not just immune response changes or acute phase, but there's really much more going on. And certainly we have papers coming out, it's not all out now, that show that there is vascular changes and neuronal changes that are going on in COVID-19, both in young individuals as well as with adults. We also thought that back then that COVID-19 positive individuals who were asymptomatic or who had very moderate clinical symptoms could end up having multi-organ dysfunction. Of course, we all know about COVID long haulers now, and maybe have been touched by that yourself or someone in your family or someone you know. So we were hypothesizing that this virus would cause this. We see it with other viruses, and we thought that would happen. So we set up immediately to be doing long-term follow-up on our cohorts. So if you go to the next slide. So this is me getting my blood drawn as one of the people who were recruited into the first study. This is the healthcare baseline study. We collected almost 10,000 individuals who work at Cedars-Sinai at multiple hospitals in about 3 weeks. And we got either their plasma draw for research or plasma draw and a mitra-tip, so one of these dried blood spots, to be able to analyze. And then we were able to provide to them, at the time was just coming out, we worked with Abbott, they released for us very early their anti-N SARS-CoV-N protein antibodies, so were they seroconverting? And we were able to provide that information back to our healthcare workers. And with that, we now have been tracking them with vaccination. We've been tracking individuals with our colleagues on people who have immune suppression or on anti-inflammatory drug therapy for clinic, and how that is changing both their COVID response, but also their response to the vaccination. So really a widespread cohort to get this all started up. Many, many, many people involved in getting this up very quickly and already collecting samples very early on in COVID. So if we go to the next slide, so what kind of things did we find out? So if you go to the next, I guess we need to advance it. So guess not. Sorry, so that's my fault. So I somehow messed up this slide, so I apologize. So what we did is, this is about 200 individuals. It's sequential time points, both healthy individuals as well as those who had COVID-19 who were admitted to the hospital, but also those individuals admitted to the hospital with suspected COVID, so suspected that they had it, but turned out to be negative. And so that's our disease control, because they're not COVID, but they're sick. And we were able to do prediction models off this and turned out to have changes. And what you can see here is you can see the healthy controls in red are really clustering. This is unsupervised clustering. And you can also see that there is a group of positive ICU patients who were positive for COVID-19 or positive with COVID, but not necessarily isolated into ICU. So they're not as sick. And it turns out if you actually look at what these proteins are driving, it's super important. So if you go to the next slide, we actually can see some of the proteins. We're actually able to pause out some of the proteins that were very important involving things, as I mentioned hormonal immune response, other complement responses, vascular response, angiogenesis. So it was super cool. And we actually have a prediction model for all-cause severity. And I'll talk a little bit about it more when we go to the targeted assays. But from the data, we're able to come up with pathways that we're able to understand what's going on, really not just in severity of COVID, but actually all-cause mortality. And of course, COVID also has a unique signature that is interesting. What we're showing here, though, is an interesting one because we're able to get plasma signatures from people who have COVID, but were asymptomatic. And we actually ended up having 2 samples. And this is with David Harrington. And we're still sorting all of this work out because it was a lot of work to get this cohort. You can imagine getting 2 time samples from individuals who were asymptomatic. So we got their sample before and then after. And so we were able to look at what those proteomes coverage and whether or not they're the same or different from individuals who were severe. One would expect that they would be, but in fact, what was quite surprising is that they had very high overlap between the mechanisms. And that implies that if you have COVID and you're asymptomatic, the main pathways are still there. Even if you go on to develop severe COVID response, that initial response that you have that is pathological is still there in the asymptomatic individuals. And I think we're not really expecting that. But the signature of an asymptomatic person is very large. And they do involve, you can see some of the proteins below, they involve, of course, things like CRP and SAA and a whole slew of proteins. And you can see all the samples that we analyzed. This is a dot one where you can see the difference if they're increased, of course, with CRP. It's an acute phase, positive acute phase. You'd expect it to go up with, if you're asymptomatic and had COVID, it should go up versus your sample before when you were healthy, it should go up. And you can see the measurements across the board. And I've highlighted 2, SAA and CRP, because that's part of our targeted assay that we did a lot more work on it. So what we found was an all-cause mortality set of markers, and as well as a COVID-specific mortality marker. So all-cause has really a pathological signature, but you have this also additional unique COVID-specific response. Now, it may not be COVID-specific, maybe viral-specific. We don't know that, but it's certainly different from having just going in for normal respiratory challenges. But there was definitely common pathways. And the common pathways are a few, but also included acute phase response. So if you go to the next slide, please. What you can see in the next slide, I think we need to go to the next one. Okay, so this is one of the big response of why we could get the asymptomatic samples was because we were using remote sampling devices. This is only one of the ones that we use, the Mitra tips, but they are very good and they're able to wick up and 10 microliters and not be affected by hemocrit differences, which is the big advantage. Next slide. So why do we care? Well, we care because diseases, and this is a modification of a time chart that we put in, Mike Snyder and I, in a commentary that we wrote for JPR a year or 2 ago, is that we really want to understand the infection, the difference between asymptomatic and symptomatic, and who's going to be affected by whatever. We also did a lot of studies on MIS-C and their autoantibody response. Long haulers, this is so critical, right? Can we predict them? And then finally, we have vaccination complications, which we're also studying. Of course, myocarditis-like symptoms is a big one, as well as blood clots, which is not surprising since we see an aggravation even with the COVID response to the vasculature. So next slide. So I just wanted to mention a little bit about targeted assays and leveraging mass spec to do these really high-end assays. This is a SYSCAPA assay. It's 11 acute phase proteins. It is essentially you capture the peptides representing each protein, and then you run the mass spec on each of those proteins. Very accurate. And some of the accuracy there for CRP is shown. really, really tight coefficients there. And so we assumed that the high level of one or more acute phase should happen at a time of administration, and we should be able to predict who would go to ICU. We weren't quite right on that hypothesis, but we also thought there might be a difference between the different acute phase proteins because they do represent different pathways. This whole idea that not every acute phase protein will be the same. SAA and CRP normally correlate with each other, but they do not have to. And in this case, in fact, they don't correlate. So if we go to the next slide, this is some of the non-normalized data, meaning this is the data that's normalized for quantitation, but has not been normalized for age, ethnicity, and sex. So we have to do it. But you can see if you look at healthy individuals versus COVID negative, but hospitalized, so there's our sick disease versus COVID positive and hospitalized. You can see, for example, CRP accurately increasing over time and SAA doing the same thing. Albumin, for example, is a negative acute phase. It goes down. And together we're able to figure out which proteins were actually responsive to give us an all-cause mortality, and also those that allowed us to do COVID positive. And again, what to our surprise was most of the proteins were not, after you take care of ethnicity, sex, and age, most of them were not good markers. So you couldn't just use them. You'd have to put in an age restriction on them, which is not what you want in a biomarker in the clinic. And so it turned out a few of the proteins turned out to be robust enough to survive all of that. And those, of course, represent unique and different pathways. And one of them is CRP, not surprising. The other one is MPO, which is really neutrophils. And I think that is true about COVID-19, right? We know neutrophils are heavily involved. It comes up to be one of the major highlights of proteins that can help predict whether or not there's a death if you have COVID-19. So next slide. So I wanted to spend just a few minutes on what's coming down the pipeline in the area of biomarkers. And this is the ECHO mass spec by SciEx. So we just got this. This is in our lab. It just got installed. We have a collaboration with SciEx for the next few years to try to figure out how best to apply this. We're super happy to work. We work with Ralph Kern and, of course, Christy Hunter, an old friend. And this is acoustic ejection mass spectrometry. So there's no LC. It's not LCMS. It does essentially direct infusion, but uses sound to move droplets very precisely and very quickly into the mass spec. And so what it does is can run up to 6 samples per second. Okay, I'm going to say that again. 6 samples per second. Now that is using metabolites. Probably a protein will end up doing 1 sample per second. But again, that's 1 sample per second. Now, the issue is you don't have dynamic range because you're not separating your peptides. For things like Ciscapa, where you've done an enrichment of a peptide, this will work really well, or we think so, because essentially we are in the last assay measuring 11 peptides. So if we're measuring 11 peptides, we could, in theory, measure 11 peptides in 11 seconds, well, about 2 seconds, because it's 6 samples per second. So you do peptide 1 in sample 1, again, go back into sample 1, do peptide 2, again, super crazy fast. And so all of a sudden, you're moving and shifting mass spectrometry to almost be a plate reader. It's going to read out your peptides super accurately. And you can see that actually on the right-hand side. This is 1 peptide being acquired 6 times a second in 6 seconds. So it's acquiring 1 peptide 1 second for 6 seconds. So it's 6 reads in 6 seconds. And the coefficient of variance was like 3%. It's insane. So all of a sudden you can start to think about how can we use instruments that are maybe not have this whole dynamic breadth, but super fast so that you can measure a lot of things very quickly. Now all of a sudden, the whole paradigm of mass spectrometry shifts from LCMS to sample prep automation systems that are able to fractionate or enrich peptides that you want to target into a system. And both can this system be able to measure peptides very accurately. You can repeat measure the same peptide and drive your coefficient of variance to be very, very small. So very interesting technology. We'll see over the next 2 years what you're going to be able to do. But for example, you could measure 400 proteins represented by 800 peptides in over 1,800 samples in 8 hours. This is just a whole other world to think about this type of paradigm. Can we do it yet? No, we're at the beginning of this journey with Christy and Sykes to see what we can do. So next slide. So what's the pipe dream? The pipe dream is to be able to move this to really changing how medicine's practiced. So today, if I'm not feeling well, I go see my physician. They may take my blood sample. It then goes to a central lab. That data goes back to the healthcare person, and then it comes to me. And normally I have to go to the lab to get the blood drawn. I don't get it done at the doctor's office. Very inefficient. So what we want to be able to do is use these remote sampling devices where the doctor does telemedicine. So they're talking to you about your symptoms by phone. You've already taken your Mitra device. It's already been sent to the clinical lab. We already know your information, your biochemistry. So when your doctor talks to you, you have not done a face-to-face with them. And this is really nice, both for efficiency, but also for accessing populations who have limited or no access to healthcare or clinical chemistry data. So you can think of remote settings. You can think of where healthcare is not easily dispersed, that going to these remote sampling devices, all of a sudden you can maybe uncouple the need to actually see a physician face-to-face and to be able to use them, let alone in the study of human population and population science and understanding disease mechanisms. All of that's equally important. But you can think about this changing the paradigm of how we administer healthcare if we could get this in place. And I believe with mass spectrometry and these devices, this is very doable. And that's what we're working towards. So if we go to the next slide. This is the development of as I mentioned before, the biochemistry, the Cedars-Sinai precision biomarker labs. Susan Marcus and Corey Bryston and Anders Berg and Nicole Leonard and I have put this together. That is the building. We are the top floor. It's just under 16,000 square feet. We have the precision biomarker labs and the mass spectrometers for the academic lab. The goal is to develop and move new biomarkers into the clinic. That's its goal. And to do that, we come up with a business plan that allows us to fund it a little bit easier than currently. And we'll figure that we'll be able to do this if we can actually really move things into the clinic and move this idea from single marker to multiple markers. And that's what we're doing here at Cedars. So can we go to the next slide? So that's our goal in biomarkers. But of course, proteomics is more than biomarkers. Biomarkers is just not even half of our work done here at Cedars in my academic lab. Usually it's about 30% of the work. Most of our work really ends up in this other part of mechanism, just like every other academic lab, working on animal models and cell-based models and human samples to try to understand so that we could go after therapeutics. If we kind of look at in that space, what can proteomics do? Well, I think there's really 4 things that it's doing now and moving out to it. One is single cell. Certainly in our lab, we're starting to work on it, trying to get human hearts and for example, in cardiac myocytes from heart transplant patients or in other ways to be able to do it to single cells to understand the population within a cell type, I think is important. It answers really biological complexity. But I would also say that proteomics is pushing to be able to quantify accurately 10,000 proteins. And certainly on the biomarker side, I think we're doing that on a high scale, seeing more and more protein. But of course, with that, you also want to be able to look at all the proteoforms, right? The chemical complexity, and there's a big push there. We're not there yet. Of course, we need to be able to do the 10,000 proteins first in the single shot versus 10,000 and all the proteome forms. And then finally, there's the throughput. You want to be able to do thousands of samples per day. And it's this balance. This is where I think proteomics tries to understand and work towards in technology development. So the goal for us is to identify and develop individualized mechanistic-based therapies which are based on a person's proteotype and molecular signatures. So today we can do protein quantity, we can do PTMs, we can look at the dynamics. So we can use AHA labeling to look at dynamics and protein turnovers, and we can look, like many other people, protein neighborhoods and protein-protein interactions, defining this complexity of what the proteome is. If we go to the next slide, but we all get stuck in having these great networks where we can see, all of these different networks are interplaying with each other. And then the question is, what do you do? Do you go after one protein, one network, one upstream regulator? And that's where we are stuck, I think, as a field, both in proteomics on the mechanism side, but moving proteomics to help us get to this individualized mechanistic therapy. So we want to be able to come up with an ability to have a high throughput method. So being able to do our goal, 96 samples in a day, proteome coverage, that represents biological diversity, represents individual genetic diversity. We wanted these, whatever we're going to do, to be responsive to perturbation, so we can knock in and knock out different proteins, do different drugs at different concentrations, just make it more. And we want to be able to track the proteome and be quantitative, just as quantitative as we are in the biomarker side, and be cost effective. So can't quite do the cost effectiveness yet, but what we're doing is the following three things. One is we're using iPSC. So our patient-derived iPSC, so we take your cells, drive them back towards stemness, and then drive them back out to organoids. We're using 96 well plates and eventually 384 well plates to put these organoids in, and then we're doing fast, robust proteomes. And so we're scaling around our patients and their iPSCs, this ability to do large-scale perturbation studies with quantitative proteomics. So if we go to the next slide. So what's iPSC? We have a large group here that does iPSCs both for therapeutics, but also to use them. So for example, if you have BRCA2 mutation, you may get pancreatic cancer, cervical cancer, or breast cancer. So we can now take the iPSC from an individual with a BRCA2 mutation and drive all three organoids, and then ask the question, can we figure out what's going on between them? And so we get to work with Clive Fenton and a whole bunch of other individuals at Cedars in this space. So we want to be able to do population-wide perturbation. So, the same iPSC, so we're going to do motor neurons, but 1,000 different people, population, or one mutation, multiple organoids, as I mentioned with BRCA2. And the goal is being able to eventually, if you come to Cedars and you have breast cancer, can we take your iPSC, derive the breast organoid, and then do the treatment on that so that we know what drug to give you first, second, and third based on your background, genetic background. So if we go to the next slide, just a few more slides. If you hit the black one again, you're going to see the I7 back in motion. So essentially what we've done to date is we grow the cells individually, we then scrape them, put them into 96 well plate, put them on the automation system, and it does the same thing: protein denaturation, reduction, alkylation, digestion, desalting. But this is not the way to go, right? Why would you make 96 different plates, either the same person's iPSCs, and then you do 96 different perturbations, or you have 96 different people, all with the same organoid type to do the study on? So this is not the way to go. So let's go to the next slide. So what have we been doing? So we've been moving to a point where we actually grow the organoids or the cells in the 96 well plate themselves. So now we're growing them in the actual 96 well plate. Doesn't seem like that should be so hard. Turned out, of course, it is. And then once they're growing on the plate, and if we've done the perturbation, we just take that plate and stick it into the I7 automation system, and it takes it all the way through, and we're ready at the end to go directly onto the mass spec because it's done the desalting. So this is our first one. We started with hex cells. We didn't do organoids, obviously, because they're too expensive to start with. One of the big issues was how to sonicate those, because you need to break up the DNA. So we worked with Cravaris to do this very cool, very focused acoustic energy that is isothermic, which means it doesn't heat up the cells. Breaks apart the DNA, does lots of other stuff. It helps on some of the denaturation, and then we could go straightly through it. So we did we go from grow the cells, stick it on the Cravaris, then onto the I7, and then the I7 does this sample prep for us, and then onto the mass spec. And we have two choices of mass spec. Of course, we can do nano. We did a lot of work on the triple TOFs, doing DIA, or we can use CE. So CE is more sensitive, so we moved to CE to see what it could do as the front end on the triple TOF. So if we go to the next slide, think it's going to tell us a little bit about, no, not yet, CE. But what we found on the Crovaris is that it was helpful. It did, compared to normal sonication, so probe sonication, not only is it better because it's 96 well plate, but it did actually solubilize more proteins, not a huge number, about 235 more proteins. This is CS data, so the numbers are slightly slower. But what was interesting, the proteins it did solubilize were proteins that were membrane associated or integral membrane proteins. So it does seem that the Crovaris is able to really loosen up a little bit more, and they were exclusively membrane associated or integral membrane proteins. So that's kind of cool. I think there's probably other ways of doing that, but it worked well for us. Next slide. So this is CE. CE is the coarse capillary electrophoresis. It separates based on electrical field. And you can, what's nice about it, it goes directly onto the mass spec. This is the SciEx CESI. The capillary is actually part of the EIS head as well. And what you can also do with CE is you can stack your sample, and you can see that here. You can put your sample in, put a spacer plug, another sample, spacer plug. So as the sample goes into the mass spectrometer, you essentially just line up your samples, and they're all in the capillary, and they all get separated on the electrocardiogram. If we go to the next one, so this is what the electropherogram looks like. This is one peptide. You can see the dose dependency as we load more into the capillary. Of course, you get a higher baseline. And you can see again, however, the protein and peptide numbers don't really change, but you do see higher intensity coming through. So if you go to the next slide. So what have we done with this? So what we wanted to know is if we were just running the CE on its own, we were getting approximately 1,200 proteins to 2,000. Can you go to the next slide? But we weren't so happy with that. So we wanted to be able to see if we could use this, the I7 to do a fractionation on a pipette tip. So we used the zip tip, and we did a fractionation at the peptide level. So you can see fraction 1, fraction 2, fraction 3. So we did 20% methanol, 40% or 50% methanol, or no fractionation. So we did no fractionation, which is what you just saw. We were able to quantify on about 2 nanograms, which is 1/10 of what we need to normally load onto the LCMS, about 1,400 proteins reproducibly. When we did the fractionation, collectively, we're able to see around 2,000. So here we're getting around 2,000 proteins. And you can do that because the CE runs are very fast, not as fast as the ECHO, but they're very fast compared to an LCMS separation. So if we go to the next slide, so what you can see here is total protein plus the overlap comparing to a normal LC run. We're getting around, on a 60-minute gradient, around 3,000 proteins. We're around 2,000 with CESI when we do the fractionation. So about 60% coverage when we were doing a 2-step fractionation. And it's a much shorter gradient time, and it's, well, run time. It's not a gradient, what you use in the CE. And we're using about 1/10 the sample. So now you know why people use CE when they're doing direct injection for single cells, much more sensitive. Next slide. One nice part about CE, though, is that it's very, very good for being able to look at phosphoproteomes. So in this study, we're moving to iPSC-derived motor neurons. So these are our patients in NeuroLinks. We did a small number. Now with Answer ALS, we're doing 1,000 patient-derived motor neurons, so 1,000 patients with ALS. We have their iPSC, and Clive here in Cedars is deriving the motor neurons, and we're doing multiple omics, including proteomics, and they're also doing a lot of clinical phenotyping and behavioral phenotyping. It's really quite remarkable program. So if we go to the next slide, I'm using it as an example. We're able to quantify these individuals because we were doing, this is on iPSC-derived motor neurons. And you can see on the, using CE, the total number of proteins or peptides we're able to get was around just over 7,000 compared to around 1,200 when we did LCMS. But again, 1/10 the amount of sample, which when you're making iPSCs mean a lot, and that's what we use in our 96 well plate. And in fact, it will be more than enough when we move to 384 well plate. We certainly have, this is very small amounts of protein requirements. But what's interesting, as I said about CE, is it is an enrichment, or it doesn't get, phosphopeptides work really well. You can see them, they're not suppressed. And you can see that here, that just on the proteome, we were able to detect more phosphosites using CE than we could when we're doing LCMS, because you don't get the suppression you see on LCMS when you use CE. If you go to the next slide, what was really cool about the ALS individuals, you can see on the electropherogram that they come out much later. Then that's in the box to them, those peptides are very far out. That's a list of the peptides, including that crazy one at the end, which is MAP1B. This peptide's massive, and it has 4 phosphorylation sites on it, and we were able to see it and quantify it very easily using CE, and we would never see that on LCMS. It's just not a peptide that would come out that you'd be able to see. Moving on to the next slide, why this is an interesting protein. It turned out that about over a quarter of the phosphosites we identified all were on this one protein. So this MAP1B is highly phosphorylated. 25% of the phosphoprotein peptides we were in this particular protein. All the peptides correlated really well, which we look at because we think it's important that the peptides representing the protein correlate with each other. Otherwise, you think there's something weird going on. The modified and unmodified proteins did a good job correlating with each other, so we were quite happy with it. To go to the next one, this is actually the sequence of MAP1B. It's a really big protein. You can't see it, but there's something like 54 phosphosites on this one protein. One of them was super interesting because we got this one peptide. You can see it listed there. It has 3 potential phosphorylation sites. We saw the mono, di, and tri-form, and we were able to see it was increased phosphorylation. That's a tri-phosphorylated peptide. It is the GSK3 beta and CK2 known site, and we thought it was important. So it was really cool to get this extensive phosphor data just coming along for the ride with the CE, which is just peptides we did not see when we did LCMS. So we go to the last few slides, which are kind of the summary. Where are we and what's the pipe dream for the targeted mechanistic based therapies? Today we have subclassification, and then you still often have to do drug 1, drug 2, drug 3, and we don't know which order to do it. It's really based on that subclassification. And we want to move from that to another paradigm. So if you go to the next slide, what the paradigm we want to be able to go, you come in as a patient, we take your iPSCs, we drive them into the appropriate organoid, we do the treatment on that organoid, see if we can find the right drug to start you with, and start the therapy we hope more targeted before going on. Can we do this today? No. Will we be able to do it? We hope so. We hope the hypothesis will work, that this is a logical way to go forward to make these mechanistic therapies for an individual. We're only at the very beginning of this road, but we're hoping it will go forward faster. So we'll see whether or not we can do this. We're building the pipelines to start to do it. So if we go to the next slide, what I'm going to say to you is, as you can tell, we dream pretty big. I encourage you to dream even bigger, but it has to be built on solid foundation. We spend a lot of time on pipelines, on quantitation, on reproducibility, on CV, on everything that we do has to meet those criteria so that we're building this strong foundation. And I think if we do that, we have a chance, if not me, maybe someone from my lab or a collaborator will be able to move forward and really move some of this stuff to make a difference in medicine. So we hope to be able to push the edge both on the technology and on the quantitation. And this is the top left is a picture from my office of the Hollywood Hills here in Los Angeles. And the bottom is parts of where I walk, where we had a big washout from a rainstorm that took away most of a road. But yet this house that's built on this edge of this mountain managed to survive because they have this amazing foundation. So that's what we hope we're doing. The next most important slide is, of course, everyone who's involved. This is a small number of people representing who we've done my labs at the ACBRI within this Smith Heart Institute. Those in red are involved in COVID-19. They pivoted big time when all of this happened. Those in green are involved really in the high throughput work that we've been doing. We run a core plus the precision biomarker lab. And of course, nothing can be done without amazing collaborators who do so much, both with understanding for us samples, technology, and that's both with academics that we get to work with, as well as our company partners that we get to partner with. So thank you.
Thank you very much, Jenny, for this wonderful talk. Before people start making some questions, I do have a couple. First, regarding your biomarker phase, discovery phase, right? I was wondering, and this is something that I think we don't actually do much, but do you consider peptidomics to do this biomarker discovery, or because we do have a lot of peptide-centric circulation systems, right? That rules a lot of things. So I was wondering if
it's a great question, and I'm smiling because I almost put a new method that we just it's actually Simon in our lab just put together, and we just presented it at MSCCL through the NIDDK workshop, just where actually for the first time we kind of approached what the question you're asking is, are there endogenous peptides that we should care about, right? And both as proteolytic products, meaning, albumin being broken down or hemoglobin being broken down, or what we're interested in is peptide hormones. So a lot of hormones that we care about, like brain natriuretic peptide, a big peptide involved in cardiovascular, bradykinin, angiotensin 2, all of these are hormones that are specifically regulated and made into smaller hormones. There's a huge number of these little peptides that are important biologically. And we don't, and they're interesting two ways. One is once they're cleaved, they're biologically active, but then they can keep getting cleaved, right? And at some point they become inactive. Some points, like bradykinin, they can actually bind to a different receptor that does something completely different. And our assays don't differentiate that. So how perfect is that for mass spectrometry, right? This is like made for mass spectrometry. And so Simon's been working on using this technology because Thermo came up with a new column, and we thought we'd try it, not realizing that turbulent flow HPLC has been around since the 60s, and metabolomics people have been using it for a long while. The Thermo column is a turbo flow. And essentially what it does, it's a combination of kind of crude size exclusion chromatography with reversed phase. So what it does is all big molecules go straight through to the wash, and you trap the smaller ones. So of course, small molecules are really great for metabolites, but small peptides get trapped too. And so you can use it to push it off because you trap it and then you elute onto a normal reverse phase. So we just started to do this. So it's pretty fun. It's like no sample prep because digestion, dilute and shoot. And we're just starting. I don't know what we're going to see. I think the big thing that we worry about is sample processing, not sample processing once it's in our lab, but the time you take your sample in the clinical lab to the time it comes and it's frozen. I think this is the scary part, right? What do you do to make sure you're not getting an artificial cleavage that's not controlled? And I think there are, of course, new tubes out there that can stop proteases, but do they stop all proteases? A lot of work to do that. But I think it's super cool, and we're just beginning. We went after bradykinin because there was a reason to do that. And we have this really cute assay we're trying to get into a paper, and we'll see. It's I think those types of technologies open up new areas. So I'd love to talk more with you, but
okay. I would love to. I work a lot with this system, so maybe we can talk later.
Oh, you bet.
Yes. So just my last question, then I'll give the time for Giuseppe and Gilberto. Maybe I shouldn't make this question now because I think a lot of people would do that. just like foresee the future. I know you are not there yet, but can you imagine how long it would take in a perfect world? The time that you take the sample, you do the IPS, you then dramatize to the organoid that you want, and then you do all these assays that you think that you should do. How long it would take for the patient to have the result?
Yeah, so it's a great question. So there's multiple parts, and part of it we don't have any control. So that's the production of the iPSC and how mature you need that particular, let's say, motor neuron. So we did a study with Clive where we did like an 18-day maturation, 32 days, 90 days. Is the signature early on sufficient in an immature neuron versus it? And, I think that field as a field is growing fast. And, I don't know how fast they'll be and how quick they'll be able to derive it. And I know some of the organoids take really long time to grow and like to develop, not just grow, but to develop into a phenotype that would be reasonable, and other ones don't. So I think there's a lot that in that field that has to mature. So it's going to be, it's not next day, right? It's going to be weeks. With respect to us, with respect to the clinical side, we hope that will get smaller, right? For us on the mass spec, what we want, we've kind of set our goals to 96 samples. So one 96 well plate analyzed by mass spec with a sufficient depth of coverage, not seeing 10,000 proteins, seeing enough representation of the proteome in a day. We're not there. But we do think if you can do that, now all of a sudden you're in a turnaround time that would be like a clinical assay. That is what we would expect from our clinical assays on a single mass spectrometer. We start to reach that proportion. I think that is more than achievable. I think there's lots of great columns out there, but it's a balance between, as I showed with the blood, coverage and speed, right? And that we have to figure out, right? And you can say during discovery, maybe we need more coverage, but maybe when we know the coverage we need, we can go down to a targeted assay, and targeted assays can be 5 minutes long. They can be 30 minutes long, they can be an hour long, but they can certainly be 5 to 10 minutes long. Now, all of a sudden, you could even ask, what's the dose? What combination can you do? I don't know how that's going to all work out, but certainly on the research side, you should be able to do many perturbations, crossover experiments, not just one pathway, but more pathways. I always think it's hard in proteomics when we do this, proteomics is broad in discovery. We see lots of interconnection. Mitochondria is talking to the lysosome, which is talking to the extracellular matrix. But then we choose one or two proteins to study as a validation so we can get the paper published. And then it doesn't go any further. I don't think this is the way to go. We have to have systems that allow us to do many perturbations, probe the interconnections of why those cells work in a certain way. And we can only do that if we have a throughput system that allows us to do it. So this is the beginning steps, I think, to really unravel that as well.
I agree with you. Thank you.
So, Jenny, I would like, first of all, to thank you for your amazing talk.
It's my pleasure.
It was really inspiring to see how actually you went from the method development and all the optimization in that part, involving even actually instrument like the EcoMS, the capillary electrophoresis, and this is really on the frontiers of new developments that maybe we will see soon coming and being more widespread in different laboratories. But also, it was also amazing to see this idea of the personalized mechanistics therapies. That is something that, we will see for sure in the future, this coming more and more, and people look into that. Regarding the sample preparation, actually, on the i7, I have seen that you talk about cell line, blood or plasma or serum. And I want to ask you, what's your experience using these robots for tissues, for example?
Yeah, so great question, because we work with some really tough tissue, like aorta. Aorta is not easily solubilized, right? Or brain also has its issues. So yes, it's a great question. So we do it, but we also put up S-trap onto the I7. I didn't talk about it here because we don't do it in these particular pipelines, but we do it for tissue. So S-trap, when I was growing up, SDS and mass spec was like you used 1% and then diluted and then tried to do it, and then FAST came up and it kind of cleaned it up and stuff like that. And now S-trap is actually, we think, works pretty well, and we use it a lot in the core. We have a little bit of discussion, is it urea and SDS or just SDS, or how do we do it? But allowing to solubilize a sample in SDS makes tissue a lot easier. You still have to crush it, you still have to get the tissue into something that allows you, a powder that allows you to then use SDS or urea. And that, I think, has really helped us to standardize method. Otherwise, we're using all different kinds of combinations of stuff. No one was really doing the same thing. We had kind of the aorta group and the heart group and the neuron group, everyone doing slightly different. Now with using SDS and then on to the S-trap has really changed that. We're coming down to really almost a standard protocol. So what I'm hoping is the S-trap group will come up with ability to use smaller amounts. I hear it's coming. So I think, so that has really, so we do that, crushing it or somehow homogenizing it without heat, then adding, I hate to say it, it's SDS. We used to use rapid jazz or this or that and all kinds of things. And now it's SDS or SDS urea. And then we titrate the SDS to try to figure out, we still don't want to use 5% if we don't have to. I think that's maybe a legacy of that. But yeah, that's what we're doing. But yeah, tissue is harder.
Yeah. I mean, and also when you talk about PTMs, and especially you focus on the phosphoprotein, but I mean, you published also several articles about citrullination. And regarding the phosphorylation, I mean, I have noticed that in your pipeline, you don't have an enrichment. And my question...
So that was just, so that's in that one. So so we have kind of two ways that we do it. So phospho, we actually, if you come to us and we're going to do phospho, we'll do phospho enrichment. Pretty much the same method that coming out of Denmark, your friend that has been around. We've used IMAC for a long time, and or titanium oxide. They both work really well for us, and so we do an enrichment. But it uncouples protein quant to the PTM, right? That's the limitation of enrichment. If we're doing acetylation, people use antibodies in acetylation or methylation. One of the things we just published in citrullination, there are no antibodies that work for IP, so they're stuck. So with citrullination, and now we just published for methylation, all 5 forms, acetylation and succinylation with BrPROT to do it on DIA. And what we do there is we make a hyper-enriched library. So we try to know all the potential modifications where they are, and then we do DIA. And with citrullination, we have a certain pipeline. With methylation, we do a 4 Dalton window because it actually physically separates mono from di from tri forms. So we physically separate in the mass spec. Citrullination, it is actually separated on the LC. Citrullination generally changes retention time, so we can use that as our parameter. Methylation, it's in the mass spec, the separation. So I think there's lots of tricks. Certainly, I did not grow up as a mass spectrometrist, so I think there'll be some people smarter than me out there that will learn how to do those types of things. But I think if we can exploit the mass spec on the LC or in the mass spec itself, we just have a collaboration with Brooker, so we have a timsTOF here. I'm interested to see how we can exploit that for, again, ways of being able to use the mass spec for separation of PTMs. We just got it, so can't tell you if it's other than we have a nice collaboration. It's fun. We're having a great group with them. So we'll see. I think all of these are really good methods, right, to do it.
And do you think, Jenny, that the PTM-based monitoring can enter into the clinic in the same way you are measuring, for example?
I think it has to. Yeah. My thing is, I think we have to. When you think about, like, if we really want to invoke this idea that we want to monitor the complexity of you, meaning diabetes and its influence on your drug, your heart disease, because that's what we're treating, your obesity, your smoking habit, and your drug will react differently depending on what you have, right? So if you actually want to measure that, you don't have enough proteins. you don't have enough proteins to be able to measure that complexity between you and me. And so to me, the way you do that is with PTMs or maybe isoforms. And PTMs, because they are dynamic, should allow you to do better and closer mapping to the phenotype. And we have some examples of that, phospho-tau is an example, but there has to be many more. The question becomes which PTMs are going to be stable enough in the blood for you to be able to use them. Is phospho actually stable enough? Well, you have to do the experiments to find out, right? To find out if the site you're measuring is stable enough. But it might turn out that's why we like citrullination. It's irreversible as far as we know. So once it happens, it has to be protein turnover to get rid of it. So if it's there, it's in the blood, we pretty much know where it came from in theory. So I think you have to think about which PTM to go after. But I think PTMs, isoforms, clearly, cardiac troponin is the biomarker, not the skeletal form. It's cardiac because it's heart. I think, people are coming up with conformational ways of looking at the conformation of a protein, like in Alzheimer's, as a way of being able. So I think mass spec allows you to get closer to the biology at the protein level. We should exploit it. That's the whole thing. Getting the marker that's closest related to the pathophysiology and the drug treatment. And if that's a conformation or a PTM, we got to be there. Great question.
Thank you.
Tiago, may I come in?
Sure,
Jenny, thank you very much for your enlightening and outstanding and inspiring presentation. I love you. Jenny, I wonder whether you looked for or you found any difference between the profile of two asymptomatic protein profile of asymptomatic and symptomatic proteins patients. I'm sorry.
Yeah, so they are definitely different. There is definitely unique pathways in a severe COVID patient compared to an asymptomatic. What surprised us, though, is that there are actually quite a few pathways that were the same, right? So there were pathways that are pathological that we saw in the asymptomatic individuals that we also seen in the severe. So it's like a common mechanism that severity that signals all the way back to the asymptomatic. But we also found some very interesting protein signatures in the asymptomatic individuals that were not in the severe. Things that were involved in the mechanism. And when I would say to you, this is what we saw, you go, oh my goodness, that just makes so much sense. So there are definitely uniquenesses that are in both, which, maybe in hindsight, we should have thought about. We didn't. We just did the experiment. But there's also common mechanisms. So I think what that tells me is that one is we should be able to screen for asymptomatic individuals. There should be a way to be able to do that. Whether or not that's cost effective or what would you do if you figure out someone is asymptomatic, I think that's a business thing, right? That's not necessarily to do with medicine. It's to do with, would that be uptake? How would it be used? You could see how it could be used, but is it, who's going to do it? There's a business component, unfortunately, to biomarkers that has to be figured out if there is an opportunity there. But I do think mechanistically it tells you. I think the common part tells you that maybe there's a drug therapy, that if someone is asymptomatic, a hypothesis could be is that if you treat them, maybe they won't go on and become long haulers, which of course they can have that, right? That's one reason we find out that they are asymptomatic is that they're long haulers, right? So I think there might be stuff there that would be of help. The question is, if COVID stays around, which I think most of us think it's going to happen, and then there's a reason to do this, right?
Yeah. I wonder whether there would be a protein that would tell us that the patient will progress to death or whether he will be able to come back, like a mortalin protein.
So it's a great question, like a prediction. So you can end up in the hospital. Can you predict who's going to be most severe and who will die? So we think we have some. I mean, and of course, this is not just us; there's other people working in this area. Okay. So I was really excited, and I told my daughter, one of them, that we think we got that. Her response, and I thought, I have to think about it this way. She said, "Mom, what does that mean? You're not going to treat them then?" And it's like, no, we're going to give them more resources. And it's going to help with understanding when you have a cost-limited healthcare system, how to prioritize things.
Yeah.
And she said,
Yeah.
Mom, think about what you just said about that at the ethics and moral level. And it's not that it's right or wrong. Of course, there's reasons that we need to do that. But it's parts that sometimes we forget about around what we're trying to ask medicine to do. When I always say we have to give physicians quantifiable data that allows them to make ethical decisions about treatment.
Yeah.
Whether or not society is ready for that, I don't know. But yeah, I'm sure we have markers, and I'm sure the field has markers like that. It is an area of research by a lot. There's algorithms out there already, but the implementation and the uptake has interesting ramifications, which is why there is a whole field of ethics and medical ethics that has to be brought into those. So great question, and yeah, there will be, and I think.
Yeah, it is a crossroad between science and ethics. We have to
always
think about it. Yeah. Thank you, Jenny. Thank you.
My pleasure.
Jenny, we have a question from the audience. It was from Magno. I don't know if you can see it. He was asking about the organoids.
Yeah, so great question. So this person's obviously worked with organoids or knows enough about them. So there is a lot of challenge around growing both the number of passages, just like all cells, they have to undergo passages, about their growing condition, how reproducible. So in the answer, so it's a great question. We kind of learned it a little bit the hard way in Neurolinks, but in Answer ALS, we have batch controls and individual controls, which means we have samples that we grew out a lot in one batch, and then we digested it and we just always take it on. That's just a technical control for us. But we also have, with Clive, the same line, meaning the same person's iPSC grown every single time. But of course, it's brand new and it's growing with the same lot all the time. So it's kind of a batch control. And then we have one where the same person's was all taken and it's now in single vials. So it's all the same passage, and they are taken each time we do a batch. So you do need to control for that. That is an issue. And I think because of that, we do a lot of kinds of QC around it. And I think it's interesting because it talks to this this, again, stretch and pull between, is it better to have a pure cell line? Heck, there's always HEC cells, right? Well, maybe not quite, but you know what I mean. Versus having something like an organoid where you are in an environment of different cell types. So one reason when we grow, for example, the neurons out later, you can see that the proteomes are different because part of it is the microglia is changing and growing as the iPSC is an older, you have more of one type of cell in addition to the neuron. So you're growing the environment around it, the cell, that environment that makes a cell, more of a neuron versus an immature neuron. So how do you kind of, these complex samples, how do you kind of figure out what that is? And that will change every single time you grow them because it's not identical yet, right? So great question. Yeah, we try to control it. We also do a lot of the same sample over and over again and the same lines regrowing to try to figure that out in any hypothesis we generate. It's a lot of work, but I think it's kind of also reflecting tissue, right? We do a huge study on the aorta, and every piece of the aorta is different. Some of it has more fatty streak than another piece, and we have to document that, right? Because the proteome is an averaging technology, right? It averages everything it sees. And understanding how to get at that. And I think that's why single cell is interesting, but single cell is now taking that cell out of context too, right? You've now isolated a cell. For us in cardiac myocytes, we have to digest all the collagen around it, get out that myocyte, which is now not the same myocyte as it was in the tissue, and ask, what's it doing? I'm actually super excited about single cell perturbation studies. I want to take my site that I've isolated, my single one here and my single, and do a perturbation study. I think that would be super cool. we can't do it. Maybe someday we'll be able to, but to me, again, because I worry what we've talked about, when you have a sample and then you have to go through this isolation or the sample processing or getting it from a patient, it's not the same. We can't pretend it's the same. It's not that it's wrong to study it, it's just not the same, right?
Yeah.
So biology is complicated.
Yeah, for sure.
Great question.
If I can make one more question, can I do one more, Giuseppe and Gilberto? Okay. So we always ask for the speakers, like, so we all know that the instruments are super fast, super sensitive, super high resolution, and we always ask, well, what's needed more for the field? So if I understood correctly your talk, and you are in the edge of the clinical proteomics, you are doing like hot rocket science there. So it's my understanding that the problem is the chromatography, right? It's not good enough for clinical science. It's not fast enough.
Well,
It's not reproducible enough, fast enough. And maybe if we can take it out and go, for example, like this team stuff and ion mobility, that the instrument does this kind of roughly separation, and that's it. This is like what you would prefer.
So it's a great question. So right now, the instruments that, we use triple quads in the clinic, and we run pretty high flow rates to keep them stable, and they're good. I mean, they're good. But you're running higher flow rates, right? And you're running triple quads. I mean, that is and that's, it works. I mean, that part works. But the question is it sensitive enough? The question becomes, well, what markers do you want to measure? Do you need to do like an enrichment? And if so, then you get your sensitivity. So I think, but it's an extra step. So I think what you're talking about is kind of the elephant in the room. Can we get to a place where we can run something like DIA in the clinic, measure hundreds of proteins quantitatively, and release a subset of those as clinical markers? So can we, for example, instead of running a targeted CRP assay, can we run DIA, have good enough accuracy to release the CRP data from that DIA run clinically? And if we do that, is there value? So that's what they're starting to do, obviously, in genomics, to be able to do whole genome sequencing and then release a subset of that panels clinically. But behind it, you have the whole genome. So can we do something like that on the proteomics side? We hope so. We hope there's value. We think there will be, but can you do that? So what are the things you need to worry about? So definitely the LC reproducibility, the columns, all of that, sample prep, on the automation system, and then you need to have the mass spec. So the question is, do you do that as multiple targeted assays or as a DIA? Having something like the timsTOF, which is now why we're working with Bruker, because they have an extra dimension. Will that help us with specificity? Meaning that when we quantipeptide, we've been able to pull things in a different dimension, right? So we have a different dimension. Will that be helpful? Will that help us for making mistakes, quantifying what we thought was a single peptide, but it's not? Will it allow us to see more of the chemical landscape? Does FAMES do the same thing, but cheaper, faster? I think these are all questions that are technically important and around, again, reproducibility. But the real question really comes down to, how do you envision these large panels in the clinical chemistry lab? And it is coming from the clinical chemistry side. What they will say, it's hard enough to do one protein. It's hard enough to make sure that you know exactly which peptides, that you have accessioned every single reagent so that you can, because someone's life is, right? It's no longer an experiment. This is real life. If you misdiagnose that person, the consequences could be huge. So now, that's why they have so many rules and regulations about every single assay that goes into the clinical chemistry around. I mean, I'm not a clinical chemist. I just listen to them and then I read their documents, and there are so many things that we have to worry about in our clinical labs here that we have for mass spectrometry. So now you make it 2 proteins, which is great because it's more cost effective. how big can you go? As you go out, I see value, but the question is, how do you do it? So we're talking about things like we will be able to measure more, but we'll only release a subset of that data that has qualified. Can we do that? What are the rules and regulations around doing that in the US? It's going to be different in different things. So I think conceptually, that's where we need to go. We are going to push it so that they can never say the technology is not good enough. We have to say, no, the technology can do it. See? But you also have to understand that the rigor to which they have to release data cannot be jeopardized. So the question is, can we reach that criteria? And I think the answer is yes. Even if it's not for every protein, but we should be able to do it for hundreds of proteins. And I think if we can do hundreds, it's the beginning. Because someone out there is a lot smarter working on instruments and LCs who are going to come up with the next way to do it, right? So I think, on my side, what we can do is push the push the rigor and the clinical utility into a place where we make it possible. But we'll see. It's a journey.
Yeah.
Can I just make a comment?
Of course.
If there are no other questions from the audience. So actually, Jenny, I would like to remember the first time I met you in person. It was during a UPO in Orlando, and it was during the early career researcher meeting. And I was already impressed by the way you were looking at the diversity of the people and be inclusive. So trying to bring everyone together, creating this environment. And I was really, really impressed by that. Because finding persons that actually are so inclusive and they allow many people from different backgrounds to participate at a different level, that was really amazing. But then, listening to your talk at UPO in Adelaide, when you were awarded by UPO, it was even more amazing because, there were other professors that got awarded, but clearly there was a difference in your talk that was the attention to people. And I remember your talk because you focused your talk to, I think, your daughter, right? It started with that, and then you went deep into something related to the attention to the patient. So thinking about this attention to people, now we have several women that are scientists that are here in the listening to your talk. So I would like to ask, what is your advice to a young woman that is now entering this proteomic world and maybe even going to the clinical chemistry world? What is your suggestion or point of view? What would you like to suggest to these young women that are here listening to you?
So first of all, I think seeing diversity is really about harvesting the smartest people and also looking at life a little bit differently. Like I said, when I asked my daughter, I was so excited. We had this all-cause mortality marker maybe, and she just kind of said something. I went, yeah, you're right all of a sudden. And so having diversity helps you to be more balanced and to see life in a way that, we get blinders on, right? That's who we are as people. So I think it helps us very much to be more better at our jobs, better at life, better at understanding. And I will repeat, I think what we do is really hard. I think science and proteomics is really hard. We're trying to change things that are just hard. And so we have to work together. My advice is probably the same regardless to men and to women, because I think also there's a generation difference in males and how they participate in life balance. And I think my husband decided to stay home and raise our children. This was unheard of when I went through. I know the battles that he fought. People just used to ignore him. And so that didn't go over so well with me. But I think what it is I think recognizing that science is really hard. We can't jeopardize excellence. Science excellence is science excellence. That doesn't mean you have the most mass spectrometers. It's how you use your brain, how you think about a problem, how you solve it, the rigorousness to which you do that science. That's excellence in science. So we have to always be there. right? And find the ways to be excellent in whatever situation you're in. And if you can do excellent science, I have to believe you will succeed as a scientist. You may not, society says all kinds of stuff about what you're supposed to be successful. I would say if you can do excellence in science, you have succeeded already. Then the rest about is how do you do that? How do you balance the situation you're in, you're both at work as well as at home, as well as your family life, as well as your colleagues, to do that. And that's the part I think you have to focus on. Now, there is definitely bias across the board, but I really do have to believe if you do excellence, it will be recognized. And you have to focus there. But you also have to, choose the battles to fight. For example, I will not ever stand up for a women's only award. That's a decision I made early on, because women should be just as good as a man. So we should be equal competition about science excellence. But each person has to do it. And I think what I missed, and maybe it was both good and bad, is I had no mentors. There was no women scientists that I was taught with in university or in where I did my PhD. There was no female professor. So that was hard because I had no one to look for how they dealt with all of this. Now, there are examples, and I think this is phenomenal. I think what it allowed me, though, is to also know that there's no rules. If there's no one to have ever done it, or so few, then there's no rules either. You just can do it. So I think helping each other, but focus on the science. And if we all do that, this whole generation will do better. I always say, everyone in my lab should be doing more than me. My expectation is that they're going to push medicine so much further than me, because I grew up as a peptide chemist looking at the smallest possible thing. And your generation is growing up in proteomics. In proteomics, which didn't even exist as a word 20 years ago. Think of what the world is for you to build on. And to me, focus there, and the best help each other onto each shoulder. You get to go higher, so help each other as much as possible. And I think that I have to believe that will be the success for everyone. And if you're a woman, you are equally worthwhile and equally of value and of equally importance in science. In our way that we look at life and how we use medicine and how we approach medicine. We all have to be at the table.
Thanks
which is why I like
Jen.
working with you.
Gilberto, your microphone is... Gilberto, your microphone.
No, I was just saying that I have two words: bear and share.
Very well. That seems like that should become an award, and the award named that and named in your honor, because that is absolutely so true. I always call them truthisms, right? That's a true statement.
Yeah.
That would be a great award.
Yeah. Thank you, Jenny.
My pleasure. Always my pleasure.
Thanks a lot.
Yes.
My pleasure. Thanks so much, everyone. Have a great rest of the day.
Thank you.
Thank
You too.
you.
Take care. Bye-bye.
Bye-bye.
Stay safe. Take care. Stay safe.
Bye-bye.